Welcome to this channel, The Art of Learning Botany. I am Dr. Nirupama Das, retired Associate Professor in Botany, Department of Higher Education, Government of Odisha. Today we'll discuss the structure and development of seeds in angiosperms. Seed is a post-fertilized product of flower in plants, and the seed-bearing plants are generally called as spermatophytes. All angiosperms and gymnosperm plants are spermatophytes. Have you seen seed? This, all of you have seen seeds while eating fruits, and daily we are taking different types of seeds in our food, that is rice, wheat, maize, and also different types of pulses. And also we are using different types of oils from seeds, mustard, sunflower, groundnut, and this coconut also. So these seeds are very important for plant and also for human society. Let me discuss what is a seed. The seed is a post-fertilized product of a flower in plants. Seed is a mature integumented ovule containing the embryonic plant in a suspended state of growth. During these post-fertilization changes, certain changes occurs, and these changes are endosperm develops from primary endosperm nucleus, and embryo develops from zygote, ovules become the seeds, and ovary becomes the fruit. The integuments become seed coat. Seeds are found inside the fruit in angiosperm, and they are called as cover seeded plants. In gymnosperm, seeds are not covered, they are open, and due to the absence of ovary, and that's why they are naked seeded plant. And here the number of seeds in fruits depends on the number of ovules in the ovary of the carpel. Seeds may be very small in size or very big in size, and very small dust particle like seeds are found in orchids, and very big size up to 6 kg weight are generally seen in a double coconut called Lodoicea maldivica. And let us see what is the importance of seeds. The seeds increase the number of plants. It helps in dispersal of species to different geographical areas. It also helps to overcome the unfavorable condition, and like the high temperature and water, and also different types of heat. It also protects the embryo from environmental hazards, from mechanical injury, and also from microbial infections. And due to the presence of nutritional material in the endosperm of the seed is also a food substance for the developing embryo, and it is also used by animals and human beings as food materials. And seeds also serves as a germplasm of the species and can be conserved. So these are the importance of seeds. Let us see the features of seeds. What main features are seen in the seed? The seeds shows the following features. Generally, it contains the embryo, and this is covered by a seed coat, and it has a dormancy period, and this dormancy period varies from hours to month and to years depending on different types of species. And in the evolutionary process of seed, the pteridophyte shows the seed habit such as heterospory and reduction of megaspore in the megasporangium and retention of megaspore also. But they could not develop true seed due to the lack of dormancy period and seed coat development. And here true seeds are generally seen in angiosperm, and in seeds develops in gymnosperms are naked here. You can see due to the absence of ovary they are open and they are said as the naked seed plants. And here in this apple you can see here the seeds. The seeds are inside this fruit and they are closed and they are covered, and that's why they are called as close-seeded plants here. So let us see the typical structure of a seed. What structures are there inside the seed? So you can see from this picture, a typical seed has a perfect seed coat and a micropyle, a opening between the integument and a endosperm and a raphe and also an embryo. And you can see here, this is the ground seed, the position of the raphe, then this micropyle, and you can see this seed coat, testa and tegment, and below this there is monocot seed, the maize seed, and here you can see the seed coat. And in this picture, here there is caruncle and it is a appendages. Beside this typical structure, sometimes some seed shows some appendages. And here in custard seed, the caruncle is seen and it is one appendage, and this appendage develops from the hilum, and this hilum is actually the opening, and here the opening of the ovule where it is attached to this placenta. Let us see these are the pictures of a typical seed. Let us coming to this structure, detailed structure of seed. We will find the raphe, which is an extension of funiculus in anatropous ovule, and during the development this raphe remains as a raphe on the seed surface, and micropyle is a small opening between the two integument and between the integument and remain as the same place on the seed as micropyle. Seed coat is the outer covering of the seed. It covers the embryo endosperm and it develops from the integument of the ovule after the formation of embryo and endosperm. In monocot plants like rice, maize, the seed coat is fused with the ovary wall to form a structure called as pericarp. So endosperm is the nutritive tissue. It first formed after fertilization process, and it is a diploid. It is a triploid tissue. And embryo, it is formed from the zygote. It contains epicotyle, limule, cotyledon, hypocotyle, and radicule. Thus, in a seed, there are three different genetically distinct constitutions. Here, the embryo is 2N and endosperm is 3N, and seed coat is the continuation of the maternal tissue. Sometimes the nucellus is fully consumed and sometimes it remains as a very thin layer, and this is called as perisperm. So let us see the development of seed. This development seed is actually how the seeds are formed. And the seed development begins with double fertilization. And you know about double fertilization. One male gamete fuses with the egg to form zygote. Then other male gamete fuses with the polar nuclei to form primary endosperm nucleus. So first, during the development of seed, the primary endosperm nucleus divides and forms the endosperm. This is the first step of seed formation, and the endosperm contains nutritive substance. And after the development of endosperm, certain extent to certain stage, then the embryo develops, and by sequence divisions, and already you have studied in embryo development, and if the endosperm is not formed, not developed, then there will be no formation of embryo and ultimately there will be no formation of seed. So seed coat develops from the integument of the ovule, and here the integument is the maternal tissue. So these are the major development during the seed formation. But certain other structures are also seen in the seed with certain plants, and these are the continuation of the ovule, and these are also known as these are such as the micropyle and raphe, and besides that some appendages are also seen. So after the formation of endosperm and embryo, the ovule get transferred to seed, and this is called as seed maturation. So coming to the maturation of seed, a perfect and perfectly matured seed determines actually the viability of the embryo and seed germination and establishment of the seedling. The seed development and maturation have some sequential, some complex sequential events, or here it takes place for a month or for years or for weeks also. Depending on the maturation, distinct changes occurs in the seed, and particularly the changes are in the water content of the seed. The water content, there is loss of about 90% of water. There is formation of seed coat, and the maturation process includes formation of seed coat. The seed coat gets transferred to a particular structure. It becomes hard and porous, and also synthesis of storage protein, synthesis of late embryogenesis abundant protein, acquisition of desiccation tolerance so that the seed can tolerate the dry condition, and initiation of seed dormancy. The dormancy is the resting period of the seed. So let us see, coming to this seed coat development. As you know, this seed coat is the outer covering of the seed, and it develops from the integument of the ovule after the formation of embryo. And here the seed coat is formed from two integument. The outer integument develops to testa, and the inner integument develops to tegment. When the seed coat develops from one integument, then it is also called as testa. With maturation of seed, the seed coat becomes thick and develops lignification, and sclerates develop. And in peanuts, the seed coat is very thin and peppery. On the seed, sometimes some scar-like attachment is found, and that is from the hilum. And the extension of funiculus on anatrophous ovule remains on the seed as raphe. And in some other seeds, appendages such as caruncle, aerial, hairs are also seen. So let us see the physiological changes. Physiological changes taking place during the seed development. This is actually very important changes for which the seed, the embryo remains viable and seed is embarked to germination. During this development of seed, these are the physiological changes, and there is increase in fresh weight, water loss, and there is a development of storage protein, and there is the increase in the dry weight, increase in starch and protein, and these are helpful for the seed germination, and there is the increase in the antioxidant level, and during the maturation there is loss of 90% of water content of the seed, and still this seed is the embryo remains viable and seed is living. And now you see there is also a particular type of hormone develops, that is called, it is called as abscisic acid, and this abscisic acid formation helps in the storage protein, that is late embryogenesis abundant protein. It helps the seed for the acquisition of desiccation tolerance and induction of dormancy. This particular hormone helps in the induction of dormancy. Such changes, physiological changes, make this embryo viable and seed empowered for germination. And sometimes question arises in mind whether seed is live, living or dead, but actually seed is living, because whenever it gets suitable environment, it germinates and that embryo develops to first to the a young seedling and then develops to a mature plant. So let us summarizing the facts today what we have discussed. The summary is seed is actually develops after double fertilization, and it is the ovule is actually the ripened ovule is actually converted to seed. The seed contain embryo, endosperm and very much close by a coat that is called as seed coat. And there are three distinct genetically distinct constitution of tissue seen in the seed, and that 2n, 3n and maternal tissue. 2n is in embryo, 3n is endosperm and the seed coat is the continuation of maternal tissue. And here the seed coat also shows two distinct part, that is testa and tegmen. Testa is little bit thicker and hard and tegmen is thin and porous. Seed coat protects the embryo and gives the signal about the environment. It gives signal about the changing environment and therefore it protects the embryo. During the maturation, the protein and starch and leaf proteins increase and there is loss of water content in the seed. Then the particular hormone abscisic acid plays a major role in the physiological changes during seed maturation. Seed also develops dormancy. Such changes in the structure and physiology occur to protect the embryo and to make the seed empowered for germination. So this is all about today's discussion about structure and development of seed. Let us see some question answer, and in the first self-assessment, these are the questions. Fill up the blanks with most appropriate answer from the multiple choice given in each bit of the question. The tegmen of the seed coat develops from dash of the ovule, and the funiculus, outer integument, chalazal and inner integument. This is the correct answer is inner integument, that is D. Then second question, when the seed coat develops from ovule with one integument, then it is called as dash. Tegmen, unitegmi, testa and pericarp. Here the correct answer is testa, and that is C. Then third question, dash part of the seed is the continuity of the mother plant on which the seed born, and dash part of the seed is the continuity of the mother plant on which the seed is born. That is, the correct answer is seed coat, and that is the C. Then fourth question, the seed bearing plants are called dash. The seed bearing plants are called spermatophytes. The answer is D. Then raphe in the seed coat is seen on the seed that develops from dash of the ovule. Raphe is the correct answer is here the choices are orthotropous ovule, anatropous ovule, cystinotrophous ovule, campylotropous ovule. Here the correct answer is anatropous ovule B. Then tegmen is dash. That is choices are embryonic shoot, embryonic root, outer seed coat, and inner seed coat. The correct answer is inner seed coat, that is D. Then seventh question, caruncle is dash, an integumentary outgrowth, scar of the funiculus, attachment of the funiculus, fleshy outer layer of the seed coat. The correct answer is A, that is an integumentary outgrowth. Then 8th question, in albuminous seed, food is stored in, and in non-albuminous seed, food is stored in dash respectively. Here choices are endosperm cotyledon, cotyledon endosperm, nucellus cotyledon, scutellum endosperm. Here correct answer is endosperm cotyledon A. The ninth question, the remnant of nucellus present in some seed is known as dash, endosperm, perisperm, aril, and pericarp. The remnant of nucellus present in the seed is known as perisperm, that is B. Then 10th question, dash part of the embryo remains close to the micropyle of the seed. The choices are femule, coleoptile, cotyledon, radicle. Here the correct answer is radicle D. Then coming to the next assessment, give very short answer to the following questions. Here the first one is, what are in 2 to 3 lines, what are the parts of typical dicot seed? The parts of typical dicot seeds are seed coat, embryo, cotyledon, endosperm, micropyle, raphe. In some seed, raphe, caruncle, hairs, and arils are also seen. Second question, name different appendages found in the seed. Here the different appendages are caruncle, aril, hairs, and they are seen found in certain seed. This caruncle is the outgrowth of the integumentary outgrowth found in castor seed, and aril is a fleshy outgrowth in lychee, and also it is edible, and seed hairs are seen in cotton, and here these are all known as the appendages. Then third question, which changes are found in the seed during its maturation? During maturation, the following changes are seen. The color changes, and the color changes from dark to black color, increase in dry weight, increase in storage proteins, and that is late embryogenesis abundant protein, and then level increase in level of antioxidant, decrease in water content during this, and also it develops dormancy. Then fourth question, write the important function of seed coat. The important function of seed coat are, they protect the embryo from mechanical damage, from microbial infection, from environmental hazards. Besides that, it helps in dispersal species because sometimes they develop some special structures like hair and wings. Then fifth question, name three major components of seed and from which a part of the ovule they are developed. The major component of seeds are embryo, endosperm, and seed coat. Embryo develops from zygote during the development, and endosperm from primary endosperm nucleus, and seed coat develops from integument of the ovule. So these are the today's discussion. I think you will like it. You share and subscribe. Thank you all for watching this video.